Okay, good afternoon, everybody. Any things that you want to raise before we start? So today we have a guest lecture, and then we'll have a student lecture, and then I'll talk a little bit, and that will be it. And next week we have another guest lecture, Paul Witwulax from Marseille, and me and another student lecturer, and then we'll have the final class, which will be only me. So, okay, so I'm happy to have here today Tal Shay, who will complement. I mean, we are a course on data science in cell imaging, but there is a whole lot of other things going on in computational biology, and I asked Tal if she can show us, present to us some of the field called systems biology, which is more mature than the field I'm describing to you. Tal is a faculty at the Life Sciences Department, and she has an affiliation also with our department. So basically, she's also a PI in our department. Okay, Tal, go ahead and you can introduce yourself more than that.
Okay, so I understand that you come from various backgrounds, so I will ask you to stop me and ask if you don't know some of the terms that I speak about. As Assaf told me, I don't have to cover anything specific, okay? So if I don't get to the end of my lecture, that's totally fine. So just stop me when you want. Assaf also told me what is the title of the lecture, okay? So I'm a bit both limited and inspired by it, because he wanted ideas from incorporating ideas from system biology into data science of cell imaging. That's the correct term? Yeah. Okay, so this is not really well defined. So what I'm going to do is first give you some definitions of the field of system biology. And I just say that the field of system biology is not well defined almost by definition, okay, because it is not well, just not a well-defined field. But I will give examples of some works which I think are nice works in system biology, representative to some level of what is being done in this not field, and I will speak about a bit about my work and mainly answer your questions if there are. Okay, so let's share the presentation. Okay. So anyone actually knows what system biology is or have some idea? No. Do you know any system biologists by name? Otherwise? No. Okay. So the encyclopedia definition of system biology is the study of the interaction and behavior of the components of biological entities, including molecules, cells, organs, and organisms. And the thing is that you study the system and not each component separately. So a metaphor I really like is based on a paper, which I'm sorry, I don't have the citation, and I probably should put it if Assaf is going to put it online. When biologists want to learn how a radio works, okay? So the equivalence of what the standard biology is doing, they take the radio and they just remove each part of it, and then they see how it affects the radio. Okay, now since you come from an engineering field, this is not an approach that an engineer would take, right? right? Gil, I said.
Yeah, it can be. I mean, you can take things apart and check how they work. It's
They
possible approach.
They just take each component. So it's the equivalent if you just take you have an electric circuit and you just take out one cabal at a time, or whatever you say in English. Okay, and see how it affects the radio, and then you say, well, the radio needs this or the radio doesn't need it. So this is how traditional biology works. You just take one gene at a time, one protein at a time, you do a knockout, and you check if the organism dies or is alive, or in bacteria, we also have the parameter of how fast they grow. And what system biology is trying to do is trying to put a bit more systematic approach into this study of the system. So let's see. So it is basically the computational and mathematical analysis and modeling of complex biological systems. And it is an interdisciplinary field because typically it's people with some biological background or coming from biology, but also have a computational background. And what's unique about it is the holistic approach. As I said, you look at the system and not at each component separately. And you ask two system biologists for the definition of system biology, and you will get 3 answers. And this is, well, this paper claims that this is because the field is young and highly interdisciplinary. But I would also argue that, as I said, it's just not one field. It is a cloud of research, I rather say. And also, if you look at the words that they describe system biology, they are somewhat contradictory. Okay, it's diversity, simplicity, and complexity. And I think here, as in many other cases, the principle is that you want the model to be as simple as possible, but not simpler. Okay, so sometimes in biology, this means complex. And system biology is really built on many disciplines in biology. It takes from the molecular biology the idea of information transfer, from physiology the importance of adaptive states of the cells and the organisms, from developmental biology the importance of the succession of physiological states, that there are many states coming one after the other which compose the biological process. And it takes from evolutionary biology and ecology the idea that all aspects of an organism are products of selection. So selection can happen at the gene level, at the protein level, at the organism level, but also at the community level. And of course, it also takes from math, physics, computer sciences, statistics, and engineering, and probably some other fields that I forgot. Okay, so now I'm going to give you some less formal definitions, but before I need to give you a background of just American history. So this is a sentence said by Supreme Court Justice when he was coming to state if a specific movie was adequate for viewing. Okay, that was in 1964 when there was still more restrictions. So what it basically says is I'm not going to define porn here. Okay, I'm not sure I can do it. I'm not sure it can be done. But what I can say about porn is I know it when I see it. Okay, you'll give me a movie, I'll tell you whether this is a porn or not. And this definition was also said about system biology. Like porn, I know it when I see it. Okay. And it was used by Mark Kirshner, the founding chair of the Department of System Biology at Harvard Medical School, which I think was one of the first departments of system biology. And still, there are not so many departments because, as I said, it's not a well-defined field. But this department gave rise to many very good researchers in system biology. Some of them I'm going to mention later. And now the next definition was not actually given as a definition, but it was stated by Shai Tal, who spent his post in the same department. And every time he was coming out of a seminar, he said, so all biologists would say, wow, cool math, but the biology is trivial. And all mathematicians would say, wow, cool biology, but the mathematics is trivial. So this is a kind of a definition of the field. And I think the beauty of it is that people can actually combine biology with math to get some insight. And my own working definition is, this is when I see cool wow research, I typically think it's system biology, but as I said, that's very, very subjective definition. So what is the toolbox in system biology? First, we have a lot of measurements because we need good data to build our models on. So those have to be quantitative and not qualitative. And very often they're high throughput. So we measure the expression of all genes in the cell. We measure many proteins in the cell. We measure whatever, okay? But the measurement is very, very important because, you know, garbage in, garbage out. If your data is bad, your model is not going to be good. And indeed, the next tool in our toolbox is modeling. And then we also have reconstruction, tweaking, and mainly theory. And system biology is not a branch of physics, as opposed to what some people think, but it does occupy many, many physicists. And Israel is really, really, really important in system biology. So I think it is one of the only scientific fields where when you will go to look for a PhD or postdoc abroad, they will ask you, why didn't you stay in Israel? You have the best system biologists there. And I think, well, we have very, very good system biologists, okay? And this is a new field. And you probably, most of you didn't take evolution course, but in evolution, you learn that there is a very strong effect, which is called the founder effect. Anyone ever heard about it? No, founder, Mia said. Okay, so it is very important when we look at a new species that is arriving at a new environment, okay? So it's not the entire species coming, right? We have one or two individuals that are coming to this new environment. Let's say we have a turtle that arrived at a specific island, okay? Well, pair of turtles, obviously. And then they have their progeny there, and then they, let's say they're very, very successful, and they end up being many, many turtles on this island. But then there was this first pair of turtles that arrived into the island. If they were, well, I shouldn't have chosen turtles because I don't know any characteristics of turtles. Let's say they were orange, okay? They happen to be orange because turtles are in all colors around brown. So in this island, all turtles will be orange just because the first two turtles were orange, okay? So, I mean, the first individuals that are starting the new settlement actually set the spirit of the settlements in human and the biology of the settlements. So as it happens in Israel, there were quite a few PhDs that finished their PhD in physics and went to do a postdoc abroad. And they did their postdoc in biology lab, and they just came back to Israel and started doing system biology. They actually started it in their postdoc. This is actually the first, Eitan, Eshel, and Elisha are physicists by training, and they moved into biology. But Ro'i, Uri, and Nama are physicists that went to a postdoc abroad and came back as a system biology. And these three are coming from Aviv Regev lab. So, and this is by no means Mematze list. So this is only a partial list, okay, of the system biologists in Israel. And now I'm going to give you two examples coming from Uri Alon's lab, and I think one example from Reiki Sholi lab. So they were all made in Israel, though some of the research was done out of Israel. So the first research was trying to answer, are biological system optimal? And well, biologists typically wouldn't think in those terms because, I mean, unless you believe in Briatanut, okay, biological systems are not designed, okay, they just evolve. And the selective force indeed set what is possible and what is not possible, but it doesn't necessarily create optimal. So like in parenthood, it's the good enough organism that survives, okay? It doesn't have to be optimal. You just have to have the most, the largest number of kids. But then they ask, do we really see optimality in the expression levels? So each protein has its basal expression level, okay? And the expression level can be, the optimal expression level can differ between different conditions. But in order to answer this question, they had to be able to calculate the optimal expression level. And then they had to be able to measure the expression level and see if it is indeed optimal. So indeed, they were looking for a system where they can do that, and they chose the lac operon. Assaf, you didn't speak about the lac operon, I'm guessing? No. Anyone here ever heard about the lac operon? No. Okay, so first, operons are structures mainly in bacteria, where you have several genes that are encoded on the same RNA molecule. Okay, so for practical purposes, it's like a gene. The lac operon has several genes which are responsible for the metabolism of lactose. Lactose, you know what that is in Hebrew?
Probably they don't.
Milk,
Sorry?
the sugar and the milk,
מה?
לקטוז, סוכר כאילו בחלב.
הסוכר בחלב, נכון. נכון, זה הסוכר בחלב. אז, sorry, milk sugar. So E. coli, which is the model organism, the bacteria, which is a model organism, it prefers glucose, okay? Like most of us, I'd say, prefer glucose as an energy source. But if there is no glucose, it uses lactose, okay? So you can understand that if there is glucose around, we don't want to turn on the lac operon. Lac operon, as I said, is the group of genes which are responsible for lactose metabolism. But if there is no glucose and there is lactose, then it does want to turn it on. If there is no glucose and no lactose, no point in turning it on because there's no lactose. Okay, so we're clear about the system? Yeah, I actually had a table, but I forgot to put it. So if both sugars are around, no lac operon. No sugar is around, no lac operon. If there is no glucose and there is lactose, then we get the lac operon. So how do we define optimality here? We can actually calculate the cost benefit of this lac operon. So there are several unique characteristics of this lac operon that enables us to do this. We can actually turn the operon on, but without having lactose around, by just using something which is very similar to lactose, but the E. coli cannot metabolize it. Okay, so we put this fake lactose, I'd say, sorry, fake lactose, and we measure how much this affects the fitness of the E. coli. Now, how do we measure the fitness? We just see how fast they grow, okay? And we can put as many of these fake lactose as we want and see how much slower the E. coli grow. And this is the cost of activating the lac operon. And we can also do the gain of the lac operon just by measuring no glucose, no lactose, how much, or a specific level of lactose, how much faster the E. coli grow. So we have the cost and we have the benefit, and they depend on the concentration of lactose, and we can actually measure that. Okay, so we can calculate the optimal level and measure the actual level of the lac operon. And what do you think? Are they the same? Anyone cares to guess? Well, it's yes or no, it's not.
Well, probably no.
Probably no. Okay, so actually I was lying. It's not yes and no. So the answer is that they are not the same, but within several generations in the same concentration, indeed, the E. coli reaches the optimal level. Okay, assuming that we don't change the level of lactose. So biological systems can evolve to be optimal. Now, it's a very specific set of conditions, okay, when the lactose doesn't change, there is no glucose, etc., but under these very specific terms. So this was one example. And the other example is actually a field on its own, which is called network biology. And this field is actually trying to take biological network, which typically looks like hairball, and make sense out of them. And Uri Alon, which did also the previous project, actually is one of the founders of this field. And I think so just a minute of what are biological networks. Can you think about a type of biological network that you know? No? Okay, ever heard about food webs? Prey predator? No. Okay, so there is Toref Nitraf, prey predator, okay, where each node is a species, and you would put edges between the species if they eat each other. Okay, and this can very well be directed because one species would eat the other and not vice versa. So this is one network. Another network, which is more medical, I'd say, is the interaction network between people. So many people study this network for each case of corona, right? The person who got positive, who he met the day before, 2 days before, and as many days before as they decided at this time point. And very often in biology, we also look at protein-protein interaction network, where each entity is a protein. And if the two proteins are near each other in the cell, we would put an edge. And in my research, I often look at transcriptional networks, where each node is a gene, and we put an edge between two genes if one is a transcription factor that activates or represses the other. So, Assaf, those are terms that you've used before in the course, transcription factors, genes.
I mentioned the genes and transcriptome, and but I, it was a side note. I never actually. So I did explain the central dogma of biology, but but we didn't get into details there.
Okay, so for the purpose of this,
okay, go ahead.
For the purpose of this lecture, what is important is that there are some genes that regulate other genes, okay? And the regulation can be positive and the regulation can be negative. So it can be done by a transcription factor, factor ratio 2, which actually make the RNA being expressed. And it can also be done by, at the protein level, by adding some modification to the protein, which makes it active, or putting it into the wherever it does it work in the cell, or wherever it doesn't, well, just putting it in a location in the cell where it can do its job. Well, okay, so is it clear, the idea of biological networks? Yeah.
So Tal, we talked a little bit about the protein networks, And also I actually showed some of my research, which is networks, spatial networks of cells, right? Trying to model groups of cells as networks. But yeah, so it's complementary.
Yeah. So your networks actually have a real location in space.
Right.
Yeah, but most of the networks that those networks don't really have a location in space, so you can actually visualize them as you wish, so that it's most informative. And now I think the groundbreaking work was defining motifs in networks. So what do we mean by motifs in networks? We mean that we have a network, we have the nodes, we have the edges, and we can arbitrarily select a subset of the nodes and look at the edges that connect. Okay, and we get some, from graph theory, you can get a clique, you can get a very sparse network, you can get many options. But what they were trying to see is within specific biological networks, if we look at subsets of 3 nodes, for example, or 4 nodes, how would their network look? Meaning we can have a clique, we can have a specific subnetwork, we can have no edges, which is kind of boring, but we can have many, many options. And they actually counted those options, and they used the network parameters as the background, and they tested if there are motifs that we see appearing more than we would expect by random. Okay, with 1 node, 2 nodes, 3 nodes, 4 nodes, I think they even reached 5 nodes. And they found out that in specific types of networks, you find specific types of motifs which are enriched. And those motifs, they are enriched in most networks of the same type. So let's say protein-protein interaction, but they are different from the motifs that are enriched in prey-predator networks. And they're different than the motifs that are enriched in transcriptional networks. And they actually, for quite a few of those motifs, they assign functional role. And so I'm going to show you just some of them, which I think are the most interesting. But this is really a big field, and each such motif has quite a few papers on it. So if you're interested, you're welcome to see. So cascade is probably one of the simplest motifs, is when you have a gene X regulating the gene Y regulating gene Z. And those cascades can be negative, where the regulation is actually repression, or they can be positive, when the regulation is activation. And now, you can imagine how this cascade is activated, how it is initiated with time. So the top component is X. At some point in time, so the X-axis is time, and this is the level of the components. At some point in time, X started rising. Okay, something make this gene become active, and X started rising. Once it reached a certain threshold, it actually represses Y, and Y starts going down because X is repressing Y. When Y goes below a specific threshold, it actually stops repressing Z, and Z goes up. Okay, so this cascade can actually change the concentration of several genes, and it can do it sequentially because we have some threshold of activation or threshold of repression in this case. Now, if it is positive, then each component goes up a bit after the other. Okay, and this can be very useful because sometimes when we start a process, we need things to start turning on, but we don't need them all immediately. So this will allow us to have each component exactly when we need it. So if you do cooking, you know that you want to put the ingredients, fast cooking, you want to put the ingredient one next to the other, and you want to put the one that you need first, you want to put it closest to you. So such a cascade actually enables you to do similar organization with genes.
And you're able to control when to start the X, when to start some gene process?
Okay, so those are motifs from a network, right? So those are just subparts of the network. And yes, there must be something that is turning X on. Okay.
And it's controllable. It's something that you can trigger.
It's something that the organism can trigger. Yeah.
Okay.
Yeah. So it very well can be the arrival of glucose at the cell. Okay, or something like that. The binding of some signaling molecule to the cell, stuff like that. Was that the question? Okay. Okay, so now we're moving to positive feedback loops. And well, feedback loops, so this is double negative. So we have X repressing Y and Y repressing X. Why is this good? What is this good for?
To stop the chain, maybe. You start a chain and you want to stop it in some time.
Okay, so for this I only need one repression. Why the double?
Maybe a control group or something like that.
Maybe a control group?
Yeah, like there's no signal here because they're both repressed. So maybe this is a good comparison for.
Wait, there is no signal here because they are both repressed. Let's say there is X. So what would happen?
There will be less Y. I mean, Y will go down.
Y will go down, and what would happen to X?
Continue.
Continue. Okay, so
Why
there
not?
is a signal. X would continue.
Yes. Until you will put more Y.
Okay, until something external would happen. But what happened if I would have X to begin with? If I would have Y to begin with, sorry.
Same? No.
The same, except I would continue to have Y and no
Yeah.
X.
Same, but vice versa. Yeah.
Yeah, the same but opposite. Yeah, okay. So the nice term is exclusive bistability, where we either have X on, Y off, or vice versa. Okay. And now if we look at the same structure of nodes, both regulate each other, except now they don't repress, they activate. Okay. And now it is again bistability, but it's a joint bistability, right? If we only start with X, what would happen?
You will get Y. You will get Y that will make more X and more Y and like a loop.
So we will have X and Y around. And if I don't have X and don't have Y, then they're both off. Okay, so there are two stable options, but now either they're both on or they're both off. And actually, like Gil asked before, there should be something that started, okay? So either it turned one of them on or it turns one of them off, and then they lock in the same state that they were in, okay? And this something can come, and then it doesn't have to stay around. They're already locked in their stable state, okay? So if you look at it again in the time, a long time, you can say that this Z actually can give a pulse, turn this on or off, and go away. And the system would remember where it should have, where it was, and would remain here until Z comes again, or something else comes again and change the state. Okay, so now we move to more complicated. Again, 3 nodes, but 3 edges loops, and those are called feed-forward loops. Why feed-forward? Well, we just feed-forward the input, okay? It goes from X directly to Z, but also from X to Z via Y. And well, they're assuming that the direction is, as I said, from X to Y and Z. Each edge can be positive or negative, and we have 3 edges. So we have 2 to the power of 3 options, and we have 8 motifs. But in biological networks, we don't see all 8 motifs enriched. We only see those 2 enriched, coherent type 1 and incoherent type 1. And this actually gets a bit more complicated. So this is the coherent type 1, because we say X regulates Z and X regulates Z via Y and activates. But then we can put an AND gate, or we can put an OR gate. Right? It can be that Z is only on if X and Y are on, or that Z is on if either X or Y are on. So let's look for a minute what happens if we have the AND gate. X goes up, okay, and turns down immediately. What happened to Y? Y goes up a bit, but then what happened to Z? Nothing, because Y didn't reach the threshold of activation. Whereas if this X pulse is longer, Y would reach the threshold of activation and would also activate Z. Okay, so what this component gives us, and this is really like a radio component, okay, you will find such a component in electricity. So it would filter transient on input signals. Another feature is that we will have a delayed on response because it takes time to the signal to reach to Y, but the off response is immediate because once X is off, Z is off. It doesn't have to go through Y. Anyone knows, well, some example of filtering transient on signals in electricity, not in biology? Ever happened to you that you pushed some button in your cell phone and it didn't work because you pushed too fast? Well, it specifically happens in the off button, okay? When you want to turn off your phone, you have to actually do it for a long while because it doesn't want you just by mistake, you press the off button and your cell goes off. So this is actually, I assume the implementation is more complex, but basically it's the same idea. You only want a signal which is persistent to affect the system. Assaf, you look perplexed. Sarah?
I just realized that you cannot hear me. Yeah. I'm interested. I'm actually, we now have a project in the lab that we're trying to look for motifs, spatial motifs of communication between cells and blah, blah, blah. Yeah. So.
Okay, so I'm going to show actually my master work just to explain how I think it can be used in image processing. So maybe I will give a better example.
Cool.
Okay, so we said this is coherent feed forward loop with an AND gate. Do you want to guess what would happen if we put an OR gate, how it would affect the system?
If the pulse for X is long enough, then it'll activate the Z, and it doesn't really matter if Y had an activation that was long enough.
Okay, but what would it filter out? Okay, and how would be the on response? It would be immediate or delayed?
It will be immediate. It's supposed to be immediate, just like just like the Y is triggered. That's how the Z is going to be triggered in parallel.
Right, and the off response would be immediate or delayed?
Well, if Y is, I mean, it can be a bit delayed because of the delay of the Y. I mean, because the Y has some more time after it is triggered, then maybe just a bit longer.
Right. So this would indeed, sorry, I should have jumped to, or I just didn't cut it. So indeed, the OR gate would filter transit off signals, okay? If the X is on and it's just being turned off for a short while, like a short electricity breakout, for example, it wouldn't be turned off. Okay, and as you say, immediate on response and delayed off response. Okay, when we have the OR, I think I'm going to skip the incoherent type 1 for the interest of time. And I'll just mention that another way of turning on the ingredients right as we need them, like I said in the cascade, remember we've seen the all positive cascade. Another way is just having one regulator that is regulate many components, but each with a different activation threshold. So once this X goes up, we slowly turn Y1, then Y2, then Y3, etc. And once it goes down, they're turned off at the opposite order. So first on, first off. Actually, in the same order, sorry. So this model is also very much enriched in biological networks, specifically in transcriptional networks. And it is called single input module. So there's just one gene which controls many other genes. Can you guess why this is risky? Let's say you're looking at an internet network, okay, where you have just one server, which is server which is connected in the entire town. So
Because
everyone gets their
Hi.
internet through the same server.
Hi. I think that the risk here is if something happens to X, then all the system is shut down.
Right. Alina,
Hi.
sorry, that's the Baba.
An old colleague.
Yeah. So right. So if all the system depends on X, then it is really risky, because if X goes wrong, then the entire system is wrong. And what we really see in biology is that nodes which are hubs, meaning that they're connected to many other nodes, are more susceptible to mutations, for example, just because mutation in them would have a greater effect. And typically, this is where we will see the oncogenes and the tumor suppressor genes, which once there's a mutation, we see a really big effect. And I'm going to skip this one as well, just saying that a very simple network can actually lead to oscillations in biology, which is kind of nice. And I would just say that network biology as a field makes use of many of the tools that are provided by graph theory, which is just a field in computer sciences, to represent and analyze complex biological systems. So I didn't mention the use of really the many networks topological properties which are defined in graph theory and can also be used to characterize biological networks. And as I said, this is an attempt to connect the network motifs into the image analysis. So there are some segmentation methods which actually represent the segments as networks. And what you see here is a multi-scale segmentation, where you actually segment the image at multiple scales. So in the lower scale, each segment is very small. But then when you go up, when your resolution actually goes down, you connect those segments based on many features. So at the lower level, the only feature that you have for a pixel is the color. But once you have a segment which contains several pixels, you can also use the, let's say, the variance of the color, or maybe you can also use the direction of the pixels with the same color, or you can actually define a pattern and connect segments with the same pattern, etc., etc. So basically, you have a network which has several levels, and each node in each level is connected to many levels in the layer below it, and fewer in just one actually segment in the level above it. So you have the same network, but at different levels of granularities. And if you look at this network, you can again look for motifs in this network. And a motif in this network might very well be object. Okay, and this object can be identified because when you do image analysis, you do segmentation that you most often want to do to identify objects. And this is just how it is applied to images of cells. So what you see here are the dark objects are cells, and the white shape around it is actually the area that the cells cleared. Okay, so this is the track of the cell, and you can do the segmentation. So you get each of those cells maybe as one segment, and then tracks as several segments. And you can, so this is actually the lowest scale, and then segments are combined and combined and combined and combined. And you can imagine that in this, the multi-scale network that will be, that represents this segmentation at multiple levels, you can identify such an object as being one object which is white with a black, object within it, which is at lower level. Okay, so this is a motif in this multi-scale network. And you can go and look for such motifs once your segmentation is completed. So basically this enables you to do the segmentation as is for whatever object you wish to identify, and then just define a motif which represents your object and go look for it in this network. So Assaf, we have 5 minutes. Do you want to talk about what you're doing in this area before I move to something completely unrelated?
Oh, so we are just starting. We are basically last week I showed you how we define how from live cell imaging we're trying to define how information propagates within networks. For example, I showed you a work from Amos's lab about from Amos Zamir from my lab that is looking at how cells synchronize to an external mechanical stimuli. And I showed you that at the beginning the cells are not synchronized, and over time they reach synchronization. And I showed you some experimental data plus nice analysis. I think nice analysis that shows that the cells at the beginning, they are aware of their local neighborhood, and then information propagates across scales from the local to the global scale. So this is what I showed you. And in this project, but also in other networks of spatial networks of cells that we're looking at, we're now trying to, Amos is now trying, well, actually he's working on his revisions of his paper, but this is what he plans to do exactly after that. Is planning to look at subnetworks, like exactly what Tal showed us now. I mean, this is our inspiration here, and trying to characterize whether there are subnetworks that are enriched in several systems, how they change when the system is perturbed, whether they have whether and what is their functional role. Maybe the easiest idea is just improving the ability to process information. So each cell has a very noisy noisy interpretation of what's going on in the environment, and maybe you can build sub-networks and modular structures that are better at processing this information. But this is our pure hypothesis. We don't know that yet. The cool thing is that since we are purely computational, we're collaborating with very different experimentalists with very different experimental systems. Then we actually have, I think, a unique access to very different systems. And then we can start asking questions, what is common between these systems and what's different between these systems and across systems and within the same systems and under perturbations. So this is where my lab is going in this aspect. And it's definitely inspired by the work that Tal showed us today. So, I mean, in principle, I showed, also another thing, last week I also showed you work from Dana's Pear lab, that she looked at the pseudo time, how to measure pseudo time from transcriptomic data, right? single cell, RNA-seq data. And then I showed you how they took this data, the same idea, the same computational idea with some adjustments, and they brought it to looking at fixed cell imaging and to characterize the trajectories over the cell cycle through this, through using similar ideas of how to build these trajectories. So I mean, I think that the field is way ahead of what we are doing, and there are many reasons for that. From what I'm doing, there are many reasons for that. Maybe, I mean, can get into it or not. And I think there are a lot of smart people who are doing very cool stuff out there. And this is also why I asked Tal if she can talk to us. And you know, and I'm not inventing anything new. I'm just trying to steal the cool stuff from everywhere and import it to our field. So, and I think
wow work.
Huh?
We are looking for cool wow work. That's system biology.
Yeah, trying to steal ideas. Yeah, the trivia. I really liked your trivial math and.
It's not mine.
Yeah. Okay, you know, it's basically the same name, just you know. So yeah, but anyway, whoever's credit, it's fine. I mean, I really like it because yeah, I'm trying computationally. We are sometimes we're developing completely new things from scratch, but it's always based. Usually, it's based on something concrete from another field, right? So we're building networks. We're using a time series analysis. We're using neural networks. I mean, whatever we need, but we're adjusting it to our data and to answer questions in our field. So, and I think this is the novelty, bringing two fields that do not talk to each other, bringing them together. This is what we are trying to do, which is, I mean, systems biology is doing that already for many years, and I think we're trying to copy that. Yeah, that's it.
Okay, so we're going to have a 10 minutes break.
Yep.
Yeah, okay, and then I'll probably take about half hour more. Does this make sense?
Yeah,
Any questions? No. Okay, so now what we're going to do is evolution. So typically, we don't see evolution. Okay, you can see it in animation, like Ayoaya, for those of you who've seen it, or sometimes other movies, but evolution in real life is something which is hard to see. And this is a very unique experimental design. I hope Assaf didn't show it to you, so that I get to show you first. I need to share my screen. אוקיי, רק tell me if you can hear me. אסף, תוציא ממיוט ותגיד לי אם אתה שומע, בסדר?
לא שומע.
מה אמרת?
לא שומע. את צריכה לעשות משהו אצלך בזה שם, בזום, שזה יכלול את הסאונד.
אתה זוכר מה זה המשהו? share sound. רגע, אבל אני אעשה לכם הקדמה.
רגע, בוא נראה שזה עושה.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white.
אוקיי, אז אני עוצרת רגע, sorry, I stop it for a minute and repeat the explanation. So Petri dishes are the dishes in which you grow bacteria. So typically they're just about this size, okay? And you put agar, which is kind of a jello, which has all the food that the bacteria needs. And you put the bacteria and they grow. Okay, so if you ever done the experiment that kids do, that you sneeze into it, or you put a clean hand and then you put a dirty hand and you see where the bacteria grow faster, you know, this type. But this is a huge plate. I don't remember the dimensions, but it's really huge. And they split it into 9 bends. And in each band, they put a different concentration of antibiotics. And antibiotics, as you know, should stop a bacteria from growing. So at the external bands, you have no antibiotics, and that's where they seed the bacteria. And then at the second band, there is a very low concentration of antibiotics. And as the bands go, the concentration of antibiotics also grow. And now we see what the bacteria do. Okay, so this is regular bacteria that supposedly have no resistance to antibiotics.
First, you see they spread in the area where there's no antibiotic up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
אלרגיות? חדש מקלינקס.
Okay, so what do you say? Have you ever seen evolution like that? So I really think this is an amazing work. Okay, there is image processing in it, but you can actually follow evolution. And it is, I think in terms of education, it's really, really convincing because you can actually see how the bacteria grow the resistance. But it also has scientific value because you can go back to the plate and you can take the bacteria at each point and you can sequence their genome and you can see which mutations evolved. And you can actually see the different points where the resistance arises, and you can go and sequence bacteria from each such point, and you can see if it's the same mutation that gave them the resistance or it's different mutation. So this is, I think, really, really amazing work. This is work from the lab of Roy Kishoni, where he was still in this department in Harvard Medical School. And Ken?
What was the time scale of this video again?
I think they said 11 days.
Okay, amazing.
Okay, so yeah, it is a bit fast forward, but relative to evolution, not much. Evolutionary time scales. Okay, so this is really one of my favorite movies, scientific movies. And it does allow for the definition of cool wow work, I think. And now I just have to find out how I present. Okay, you see my presentation again? Yeah. Okay, and this is the paper. It was published in Science. It's very nice.
People don't seem excited enough. I mean, I think I showed that video to all my family and to my kids, and you know, it's amazing if you think about it.
I agree, but you know, every person shows the excitement differently. So I believe they're secretly excited. I think if Tach is happy, you can see some excitement.
It's amazing and frightening, actually, when you think about what this antibiotic is supposed to do.
Right.
It's frightening.
Especially frightening if you think about the coronavirus and how we actually, you can look at the vaccine as a type of resistance, right? Because there's a specific vaccine or 3 of them, and all the virus needs to do is actually to learn how to bypass this specific 3 vaccines. And it does have a lot of options because it still runs in many, many countries, both with vaccines and with no vaccines. And there was actually a show of concern letter that was saying that what we are actually doing is developing resistance to the vaccines. Especially, I mean, in Israel, the vaccine process was pretty fast. So luckily we didn't have resistance, but when you do the vaccination very gradually, you actually let the virus time to develop resistance. And this is what we are afraid of. And this is why I think we should give vaccines to everyone, even if the US pays for it, because it would be much cheaper in the long run than having viruses which are resistant to the vaccines. But that was my take. Okay, this doesn't have anything to do with system biology. Well, maybe some. Okay, and the C. elegans cell-fate lineage. So I would say this is not as cool video, okay? This is, well, the C. elegans, first, is a nematode. So it's not tolat, okay? But it's very similar in all practical purposes, 1 millimeter long. The nice thing about it is that development is always the same. So the cell lineages starting from the single egg and developing into the adult are always the same. So the embryo always consists 558 cells. The adult always consists 959 cells in the hermaphrodite, and the male has a bit more cells. But it's always the same number of cells. So the development is completely deterministic, which is not the case for, let's say, human, okay? And since the egg and the animal are transparent, you can actually follow the cell divisions and see if cells migrate from a specific location in the animal to another location. And then someone chose to do his PhD following those cell partitions and migrations. And he actually spent many, many, many years in front of the microscope. And this was the end result of his effort, okay? It starts, so this is the cell-fate lineage of the C. elegans. It starts from 1 cell, 2 cells. Here is the next separation. And you can see all cell partitions that are needed to create a C. elegans, an adult C. elegans. As I said, manually produce the first complete and reproducible fate map cell lineages. Okay, so whenever you say how much working with images is, I'm trying to think of a word which is not too harsh, boring, okay, think about them and feel very lucky. However, it did get them the Nobel Prize in 2002. And I mean, this is not just a tree, okay? It teaches a lot about the development of the organism. And actually, today it would have been much, much easier to produce. So I'm going to show you the movie of the zebrafish embryo, which is actually depicting very similar tree, but was done much faster. And you can see that here also you can follow the division of the cells. And they also had some markers, so they mark specific cell lineages. And you see that some markers light up now in yellow. And basically using image analysis software, I think you can create the cell fate map much easier today. So we have progressed a bit.
By the way, Tal, I talked with some people who do this type of lineage analysis today, these days. Every single trajectory is validated manually. So you have automated tools, but eventually, you know, if you have, you cannot allow even small errors, so everything is eventually manually validated in these studies.
Yeah, but manually validating it on a computer screen where you already have the link is much, much easier than creating the tree yourself, I think.
זה נכון.
And still, I wouldn't choose it as a PhD, just saying. Okay, and now we're not leaving the C. elegans, we will go back to it, but we're moving to single cell measurements. So I understand Assaf already did some scooping, not scooping, what's the word? Already told you a bit about it. So if I'm saying the same stuff, just tell me and I'll run, okay? So we can now do measurements at the single cell level. And this can teach us a lot of things. And we can do DNA sequencing at the single cell level. We can do RNA sequencing at the single cell level. And we can do ATAC-seq here is just an example, but we can basically sequence everything in the single cell level. And there are methods that would allow you to see where stuff is bound to the DNA, or where proteins are, or where the DNA is open and waiting to be transcribed, or which pieces of DNA are near each other. So what is the three-dimensional structure of the DNA? And really there's a new method of sequencing coming, published every week, I think. Not sequencing itself, but new information you can get from sequencing. So I'm just going to show you two rather representative works, I think. So one is following the clonal structure of the tumors. So you can think of a tumor similar to the bacteria that we saw before. It starts from one cell, which has some mutation. This cell partitions and give rise to another cell with more mutation. And if this mutation is helpful in terms of the tumor, it is related to האוכלוסייה, and then you have other mutations coming up, and at the end, where we identify the tumor, it is already a big mass of tumor cells, but each of them belong to a separate clone. And we can actually, when we sequence the DNA of the clones, we can actually build a tree of the clones and see, not always, but if we have a big enough sample, we can see how the tumor evolved, and we can see which mutation actually, by calculating the frequency of cells in each clone, we can also see which mutation was more helpful in terms of helpful for the tumor, letting it grow. So there are many works of this type which are being done these days. By the way, this doesn't have to be tumor. Okay, we can also learn similar to the C. elegans work. If we take cells from the normal human, and we do the same analysis, so mutations are not as often. But if we look at the sites of repeats, which are more mutated than regular DNA, we can also identify, we can also build the fate tree of the human. Well, it's not as deterministic, but we can also build it. And this was done by Shapiro in the Weizmann Institute quite a few years ago, actually, before the single cell here. And single cell RNA sequencing, which is what I do for a living, mostly. And I chose the example of the C. elegans, okay, because we just saw it. And what you see here, each tiny point is a cell. And we put the cell on a two-dimensional representation. And for each cell, we measure the expression of all the genes in the cell. And we calculate the similarity between those cells. And this 2D representation, the UMAP, is similar to PCA in sense, for those of you who know it. It tries to maintain the similarity structure between the cells, but on two-dimensional. So you can see that all the cells which are stem cells are together, and all the muscle cells are together, etc. And also, I understand that Assaf already talked to you about the trajectory analysis and pseudotime. So you can see that those cells are colored by the estimate embryo time. Okay, how much time passed between the egg and this specific cell. And you can see that the egg mostly starts somewhere around here, the dark blue, and then it develops into specific differentiated cells. So you can actually, from this representation, reconstruct the fate map that we've just seen. And indeed, not surprisingly, it matched very well. So here you actually have 86,000 single cells of C. elegans. The correlation between this and, well, I mean, it really matched the map that we've seen before. And also it gives them some more insights about how deterministic a cell fate is and how correlated. I mean, because some cells can actually come from several lineages, and they see that the differentiated cells coming from different lineages are very, very similar. But along the lineage, they also see similarity. So it's only the differentiation stage that bring those cells to have a similar transcription profile. And some challenges in working with single-cell RNA sequencing data are first that it is very, very sparse today. So we only have very few, I mean, if we have like 30,000 genes in the human genome, typically in a cell, we will have less than 1,000 genes that we measure. And most of them are only measured by one read. So this is a very, very sparse data. And it means we need imputation, and we need to correct for many, many biases inside. And then there is the challenge of visualization. So dimension reduction, like we've seen here with UMAP, is very, very common, but it is also misleading in some sense, because it is two-dimension and we're talking a 30,000 dimension space. So necessarily we lose a lot of information. And this is very obvious when you try to cluster and classify the cells.
I have a question here. What about trajectories that are not driven by the main variability in the data? So they are not driven by the dimension, whatever dimensionality reduction you are working with.
You mean that there are several processes in parallel happening in the data?
The variability in the data can come from, you know, from different origins and from different, you know, functions. And if you are interested in a biological process that is not the main axis of the variability within the data, you might not even be able to see it in you know, in unsupervised dimensionality reduction.
Right. So there are some known sources of variability which are factored out. Like you can remove the cell cycle component, okay? Because you know that cells go through the cell cycle and this changes a lot of genes. So you can actually, well, less or more accurately remove this factor. So we often do it in PCA, which is, as I said, similar to UMAP but cleaner, where you define the dimension. Okay, so one dimension is, well, one source of variability, and then you can say, okay, this source of variability I don't care about, and then you remove it and you look at the next dimension, and then you say, okay, this is more interesting, and then you look at only the second and third dimensions. Okay, but this works for, I mean, PCA doesn't work well for single-cell RNA sequencing typically, just because the data is so sparse. So you put it on a PCA and you see nothing in most cases. But that's why I say that those dimensionality reduction methods are actually doing some stuff to the data which you're not always aware of and is not always representing it optimally. So just a point to keep in mind. Okay, just to explain how sparse it is, when we were trying to identify the sex of the cells, okay, which is, a very big factor in biology, but we were able to identify the sex for 50% of the cells in a typical experiment. Okay, so there's a lot of information that is just not there because of the method. So there are some very interesting questions that we can answer using this data, but there are some questions that we just can't answer because it's not deep enough. Okay, so as I said, the quality of the data and understanding the limitations of the data are very, very important when you're doing modeling and system biology. And trajectory analysis, I'm not going to talk about because you already talked about it. So I just wanted to mention what I do for a living and for fun. So one thing I already mentioned is we are studying male-female differences in the immune system because we know that females are actually better at handling all kinds of infections and diseases, including the corona, by the way. But they are also much more likely to get autoimmune diseases. So we know that there are immune differences and we're trying to find them. We also look at the dynamic of immune responses and at splicing dynamics. And we do most of this by looking at RNA sequencing and single cell RNA sequencing, and also a lot about genomes and their annotations. So it's a lot of biology, but it's also a lot of data mining, I'd say, and data sciences. So if this is of interest and you're thinking about your next career stage, you're welcome to talk to me. I just realized I didn't put here the email, but I'm sure you can find it online, or you find the other Shaital, which is fine as well. It will reach the same place. I just wanted to mention one huge project, which is close to my heart and also to system biology, which is the Human Cell Atlas. Okay, this is collaborative effort of many, many labs across the world. I think it's the largest one since the Human Genome Project. And the idea is to profile each and every cell type in the human body in health and disease. So this is very, very ambitious. I would say it's not even well defined, but data sets are already out there. Not all of it, obviously, but some data is out there, and I think there are huge opportunities there for system biology work. So I want to thank you for staying with me, and I'll be happy to take any questions. Questions? You're shocked. Well, at least you know what to answer the next person that would tell you why you believe in evolution. And you say you don't believe in evolution, you just know it's true because you've seen it. Okay? Okay, so thank you. I don't need to stop the recording, right? You're continuing from here?
Right, we are continuing.
Okay, thank you very much. And it was good talking to you. Bye-bye.
Bye. Okay, so I think it's exactly on time for the student's presentation, where we'll see also some other types of motifs, right? And we'll see something that we've never seen in Tal's talk, temporal information, real temporal information, not pseudo time, but really information in time. So, okay, and Nadav and Yael, are you ready?
Yes, let me share my screen. second. Okay, can you see the right screen, the large one? Okay, perfect. So I'm gonna start now. So hi everyone, my name is Yael and my colleague Nadav is going to present with me a paper called Codex, which is a neural network approach to explore signaling dynamics. And it's going to connect really well to the presentation we've just seen. So a little bit of background, I must admit, I had to go and understand the background myself because otherwise I wouldn't have understood the data, and I don't come from the biology area at all. So just a quick overview of what is biosignaling. The video that you're seeing here is the cell membrane, or in Hebrew, chromata. And imagine there's a material or growth factor coming from the outside of the cell. They're going to activate some type of gate on that cell membrane, and then they're going to send some signal into the nucleus of the cell. And what we're going to focus in this experiment, or the paper that we're presenting here, is the process of a material activating this gate, and we're measuring the amount of activation. Okay. So the experiment was held in the following form. There are several types of cell, and specifically we're interested in two types of pathways that are going to be activated. And each and every time we're going to input a different type of material, we're going to therefore stimulate it and activate those pathways, and we're going to measure the amount of activation over time. So the end result here is this time series graph that you see here on the right of the two different pathways and the level of activations that those pathways had. So here we're going to call it trajectory, much like what we've just discussed in the following hour. But I want you guys to remember that the data input here is a time series data of the levels of activations of each pathway in this experiment. And then what they did, at least to me as a computer scientist, is pretty trivial. They decided to train a neural network, and specifically a convolutional neural network, for a multi-class classification problem. And what they did is that they took the labeled data that they have, and they know which type of material formed what type of data. And during this training process of the multi-class classification problem, they are stating that they found a really great latent space, or an embedding, for this type of data that we can actually explore to understand better the nature of this type of data. So essentially their entire contribution here is we are taking data that so far we needed to do some manual feature engineering, and we're replacing it with features that are generated by this convolutional neural network. So imagine that we took this type of data of activation over time, we fed it through the neural network. Their other contribution is, how do we explore this latent space? So imagine taking all those data samples in the embedding in that space, and you're doing some type of dimensionality reduction, for example, t-SNE. And here you can see on the right, at the upper right, an example of how t-SNE visualization is going to look for this data. And then we're going to do some type of clustering, which again, I think is pretty trivial for our world. But the interesting thing that they found here is that they are able to do two more steps to explore this landscape that is very meaningful to the signaling world. So the first one is, we're able to find prototypical samples, and I'm going to explain in a moment what it means. And lastly, this enables us also to identify motifs, much like the motifs we've seen in the last hour, but a little bit different, and I'm going to show how. So the first stage that I just mentioned is extracting prototypical samples. So as I mentioned, their system is built on doing a multi-class classification problem, which means that at the end of the day, the network is going to output some type of softmax vector, and there's going to be a probability of each class. So we can select the samples that receive the highest probability for the class as the top prototypical samples. So imagine these are the ones that the network is most certain about. And you can see them marked as those little diamonds in every class. And then they want to understand what is the cluster or the hull or the borders of the cluster for that specific class. So each and every time they're going to look for the most further away samples in each class from the prototypical class. And then you can see those thresholds or those borders of clusters. And essentially what they're showcasing here is that they're able to understand what type of clusters are in this embedding that are appropriate to the classes that we saw in the experiment. And Nadav is going to show it on several types of data in a moment. After they have the specific samples for each and every class, they did another really interesting thing to explore this landscape, and it is to extract the motif, meaning they're looking for a pattern that would describe this class or this type of samples that were clustered together. And the techniques that they're using here, again, which is based on the fact that they have a neural network embedding, is a technique called CAM, which is a class activation map. And essentially the idea here is to take the sample and feed it through the network and check which type of neuron or what type of specific features in the data were very meaningful towards the classification. And therefore, we understand that this is very meaningful for that specific class. So here I gave an example, and this is actually taken from a video that they generated for presenting this paper. On the left, you can see an example trajectory. Okay, this is a specific sample in the data set, and you can see that activation levels over time. And here on the right, the red dots represent information that the CAM technique said was very meaningful toward classifying this sample. So you can see, so that if you only take, for example, the red dots, you can understand that this is a specific motif, or this is the specific dynamic that we are looking for to say it belongs to a specific class. So essentially, we can understand that this is some type, I would like to think about it as a signature on the phenotype that we're actually observing in that dynamics class.
So let's talk about the experiment for a bit. The first thing that they did, which is very cool, they generated synthetic data. Each trajectory had exactly 4 peaks, either a truncated peak or a full peak. As you can see on the above chart, this is the trajectories created by the different amount of peaks. And what they found out that the amount of full peaks was extremely discriminative. As can be seen in Fig B, only the amount of peaks managed to cluster the cells pretty well. Additionally, they were able to extract motifs, as Yael mentioned, from the synthetic data. They used the synthetic data also to train the model and to use it later on real data. They generated 10,000 trajectories, 70% for training and 30% for validation. Next, they compared the model to the classical methods, the method that were used in previous experiments. This was created using a known library to generate over 1,000 features from the time series. On the left, they tried to cluster using those features. As you can see, it's clustering less well than the actual CNN. However, they did not quantify how much the CNN outperformed these classical methods. Additionally, they extracted motifs, and as you can see, it is much more scattered and less informative than the previous motifs. Again, they did not quantify how much. And last, they used a random forest for classification, and they mentioned that the CNN outperformed him because he had trouble utilizing correlated features. Again, they did not quantify how much the CNN outperformed, and they did not try to handle the correlated features, such as removing correlated features or feature selection. Last, they tried using the model on out-of-the-box datasets, on new datasets that the model did not train. They compared it to the known methods, which you can see on the left. And what they showed is that the CNN clustered the features and the cells much better than current methods. But again, they did not quantify how much. So to sum up, we would like to share our opinions about the paper. First, the proof of concept using the synthetic data is very impressive. It is not trivial. to generate data and to train on it and actually get good results on real data sets. This is very impressive. And the second main contribution that Yael has mentioned before is utilizing neural networks and making the handcraft features redundant using data-driven feature engineering. It's very impressive and very much what we are trying to do in this course. Additionally, the model can handle a lot of data and it works very well out of the box. However, we do have some concerns about this paper, mainly about the model selection. They use CNN on time series data. The state of the art currently is LSTM, transformers. Why they use CNN? They did not specify and they did not do any comparison. They talk a lot about the embedding and the latent space. So why not use autoencoder to try to explore that space? Again, they did not specify. And lastly, all the comparisons were not methodological enough. They were not quantified, mainly with the classical machine learning and the random forest. As we said, they just said that it wasn't good enough. No numbers were presented and no attempts to actually optimize the random forest were used. Thank you.
Question, thought. So they had also some data, their code and data. You told me that it was also publicly available, right? So
yeah,
it's also a nice contribution. So I know that Yael is planning to do her project by basically trying other methods on this data. So yeah, I mean, data sharing is very useful, and I'm hoping to have a lesson on that before, at least part of a lesson on that during this course.
I imagine that would make this paper really cited. For example, in the cybersecurity field that I come from, when you publish a data set, that makes your paper the most cited in our field ever. So I'm guessing this is probably going to be a well-cited paper for that reason.
Don't count on that necessarily. So there are there are small numbers of, maybe I'll just briefly mention that. I'll talk about it more, hopefully, if I get to give the talk on data sharing and reuse. First, there are very few data scientists currently in the field. So this is one thing. I mean, the data sets are very virgin in terms of people are not playing with it, are not trying to find patterns within it, which is, it's also an advantage, but it's, yeah, I mean, you're not getting tons of citations on these types of data, at least yet. Yeah, and second, it depends for what purpose. So tool development and showing, yeah, I can use the tool and improve performance. Yeah, this is now common. People do it on public data sets. Again, it's very, there are these very few data sets which everybody tries to use to, for example, segmentation, then there is these challenges that everybody tries to improve, like Tammy told us about segmentation tracking. So usually it's more standard tasks in bioimage analysis. The idea of, so this is for methods development, the idea of taking old data and finding new patterns within it, so learning something new about the biology, this is an idea that is actually quite new in cell biology. There is resistance to that for some cell biologists. For some reason, they are looking for, they need an experiment. They need you to do an experiment in order to make it worthwhile. So it's kind of an old way of thinking. I mean, in many other fields, we're not there. In cell biology, I think it's getting now in the last years, it's getting better. And I see in the sprouts of nice paper that use all data and are still, but I can give you from my own experience where I got from reviewers. They wanted us to redo experiments that were already available out there. So So, but you know, this is the struggle of a new interdisciplinary field, I think. So there are different cultures, and this will be solved. Other things. What do you think about you talked about motifs? I deliberately asked you to present after Tal because I knew she's going to talk about motifs. What do you think about now that you know Tal's motif? What do you think about the connections or disconnections between what Tal showed, the network motifs, and what they define as motifs?
So I think I almost asked her a question because it was a little bit different than what I understood from this paper. But what I understood from Tal was that the motifs there are structural, right? She said, we're going to look at the structure of the network or the graph and understand if there's a specific characteristics of the structure. While here, we're talking about a motif or a pattern that is reoccurring over a time series data. So I even Googled the word motif to make sure I understood correctly, but as I understand, it's generally detecting a pattern that is reoccurring, but the pattern can be, I guess, either structural or timely. That's at least how I understood the difference. I hope I'm understanding it correct. Actually, I would love it if you could clarify.
So I'm not sure that I understand what you're saying. So you're saying that basically it's the same thing. These are patterns that appear in the data, and these are patterns that appear in the data, only that the data is different, or something else?
So almost that's what I mean. I think that also it's patterns that are describing a specific phenotype or a class, but I think that's how I understood it, but the types of data is different, yeah. So the first one that she showed was, I think, structural one, and this one is time series one. That's how I understood it.
So I think that when looking at the network motifs, if you're doing it data-driven, in a data-driven way, you'll first look at you'll define network motifs. You look at their expected probability, and then you'll see, okay, these network motifs are enriched. And then you'll try to reverse engineer to understand what do these components mean, right? I think. I mean, I'm not. Maybe you should have asked Tal. In your case, it's a little bit different because you are defining the motifs based on their contribution to a classification task. And basically, maybe you have an implicit, right? Implicit assumption that if a motif is important for a task, then it is enriched also in the task. But I don't know, they didn't test it, right? I mean, they probably did kind of, not directly, also kind of, it probably comes under the hood, I mean, I assume. So if this is true, if this assumption is true, I mean, it needs to be validated, then I would argue that it's similar, that just they define the motifs in different ways that are more targeted to a specific function, a classification task. Now, I have a question about how did they define them scores and technically. So what did they do exactly? How did they decide this is a motif?
So they, as I understood it, they took the samples, the prototypical samples from the specific class, and they fed it into the network and then used the CAM technique to understand what, let's call it input information, was the most crucial one for, or activated for determining the specific class. So you can, when you have a feed-forward network, you can actually feed the data in, see what type of neurons were activated in the decision, and then project it back on the original image or the original input.
So what is the CAM technique?
The CAM technique is using attention, and the attention is basically focusing on specific parts of the input, increasing the value, and that's how it's signaling.
I think the word is perturbating. אני לא בטוחה איך להגיד את זה באנגלית, כאילו שמזיזים קצת את הקלט כדי לראות...
Perturbing, okay, yeah. This was my guess, right?
כן.
Okay, so this was my guess.
I didn't know how to say it in English and I was Okay.
Perturbing, yeah. So I think it's in biology it's perturbing. I hope that I see that it's perturbing also not in biology. So this
Yes,
was
also
my
in math, it's just a difficult word, that is.
so this is what I was assuming. I mean, I wanted to ask you because I was not sure. But if the idea is okay, let's take a time series and then we go to a part in this time series and we perturb it, משנים אותם, right? To something, we take some other part of the time series and we replace or something like that. And then we can see how well it is classified, right? How the classification score is reduced or increased.
Exactly.
I think this is pretty cool, right? I mean, I think it's pretty cool to understand the functional role of and what are the key properties. And then if this is true also, it's, yeah, it still doesn't mean that it is enriched because it's smarter than that. You can have a motif that is rare, but it appears in every time series of a specific perturbation. But now perturbation is the biological perturbation, right? It's the AGF, the treatment, and it will still be the driving feature for the classification task. In principle, I'm not sure what happens in this system. It could be a project, right? Of course, project. I think it's pretty cool. I like the idea of, well, you know, I'm also biased to the idea of motifs because I'm thinking of the similar thing just in multicellular settings. So I like it. Any other questions? So I had one last question. Can you explain what is this full peaks? The simulation data, I just stared at again, I didn't read it carefully, or, you know, I just skimmed. What is the full peak or not full peak in the simulated data? Because it looks, I see their peaks. I mean, what's going on?
Okay, the best thing is to go to the last part of the experiment, the paper. They explain it. It's pretty complicated formula, so it's pretty hard to explain. But basically, think about a Gaussian peak, and then they truncated and truncate. I'm not sure I'm saying it right, but they truncate one of the sides and make it less Gaussian and more steep. It's a mathematical formula. It's pretty long, so it's pretty hard to explain. But basically, this is the intuition, and that was the most discriminative feature that they found.
I think I would like to add, and this is actually something that I wasn't sure why they didn't explain. Maybe it's obvious for biological crowd, but I think, as I understand it, they tried to mimic how data would look if they actually had real data. And I think they are claiming that usually it has some type of activity that has peaks. So I'm not sure why they chose 0, 2, 3, 4. I'm assuming it's connected to the biological event. But as I understood it, they're claiming that sometimes it's a full peak, meaning it has 2 tails, and sometimes it has 1 tail or 1 end. So as I understand, they are sampling from the Gaussian distribution and generating those peaks in a different ways. And they are claiming, and I think they actually showed it pretty successfully, that when you have a few peaks, but some are truncated, they're going to be clustered in a different cluster. So I think they are trying to mimic the real activity, but again, they didn't give the biological intuition. So I'm lost as the intuition why.
Okay. Okay, sababa. So let's go to a 10-minute break and get back at 15 past 4, okay?
Thank you.
אסף?
כן.
אפשר שאלה לגבי הפרויקט?
כן.
אני פשוט הסתכלתי על הדאטה סטים שכאילו פרסמת, ולא כל כך הבנתי איך אנחנו אמורים לבחור שאלת מחקר לפרויקט. כאילו נגיד בחרתי את האטלס פרוטין.
שמעת את ההרצאה שלי על זה?
כן, כן. הייתי בהרצאה וגם שאלתי שאלות.
אוקיי.
אז אני פשוט לא כל כך מבינה איך כאילו לבחור שאלת מחקר פשוט.
זה חלק גדול מהבעיה, זאת אומרת, זה חלק גדול ממה שאני מצפה ממך לחשוב עליו ולהגיע עם רעיונות. נתתי כמה רעיונות שם בהרצאה הזאת, והנה, עכשיו נתתי נניח עוד רעיון, כן? אבל כאילו, זה המשימה.
של לחקור את המידע ולהגיע לאיזושהי שאלה שאנחנו רוצים לשאול על המידע? זה כאילו הכוונה? כן.
כן. יש כל מיני דברים שאפשר לעשות, אבל אם את רוצה, את יכולה לשאול שאלה יותר ספציפית, אז אולי אני אוכל להיות גם יותר ספציפי, לא יודע.
לא, פשוט הסתכלתי על הדאטה סטים שיש שם, שהם כאילו הם נראים מעניינים, וזה גם מתקשר קצת למה שטל דיברה עליו בהרצאה שלה עכשיו, כאילו על נגיד אבולוציות ודברים וזה, אז...
על ה-Human Protein Atlas?
כן.
אני מקווה שאני אספיק לדבר על זה, אבל אם כן זה יהיה ממש בשיעור הטוב, בשבוע הבא זה השיעור הלפני האחרון, נכון?
כן.
וואי, נגמר לי הסמסטר, כאילו. לא הספקתי, אני אף פעם לא מספיק בקורס הזה את מה שאני רוצה. כן, אז ה-Human Protein Atlas, את יכולה לחשוב מה יש לך שם בסופו של דבר, יש לך שם ערוצים שהם כאלה קבועים, ועוד כל פעם ערוץ נוסף, נכון?
כן.
אז את יכולה, השאלה אם יש מספיק דאטה, את יכולה לחשוב על איך, על המיפוי, נניח ללמוד מיפויים, מערוצים מסוימים לערוצים נוספים, או כל מיני, סתם, זה הרעיונות שסתם קוצים לי לראש עכשיו, לזה. אפשר לקחת גישה חישובית לגמרי ולנסות להתמודד בתחרויות שם שיש להם, לשפר את הלוקליזציה, את ההחלטה מי זה, כאילו בהינתן חלבון, לאן הוא הולך. זה המשימה שהם עושים. אפשר לחשוב על איך אפשר להוציא מזה משהו אחר קצת, אולי איזשהו נטוורק, איך אפשר מתוך הלנדמרקס לחזות איפה הולך להיות חלבון מסוים שלישי. אני לא יודע, אבל אם יש לזה מספיק דאטה, או אורגנלה שלישי. כאילו יש הרבה חלבונים שמגיעים לאותה אורגנלה. סתם זה רעיונות שדומים קצת למה שאני עושה במעבדה, מחקר במעבדה, אז זה ישר עולה לראש, אבל אני לא יודע.
אוקיי, תודה.
Okay, so I want to spend the next 45 minutes to talk to you about, I started last time, I'll start again, but I'll go quickly, about an example of a screen that includes within it other ideas. So how going further with the screen and using temporal information and asking a specific biological question. So I'll describe a project from this project was published in 2017 about the collective soil migration, and I think you'll see a few things that will be relevant in terms of screening and others. So the research question, and as you know, I'm still interested in that, in cell-to-cell communication, but in this specific case, it's how cells transmit information when some cells know the information and some cells do not know the information. And in this case, you'll see cells that are migrating, and the cells at the front, they know that there is an empty space, and cells at the back do not know that. So when these cells knows where it's supposed to go, how this information propagates to other cells in the group. So this is the experiment. It's called the scratch assay or wound healing assay. These are basically lung cells, epithelial cells. So these are cells from human lungs, and you can see that they are grown to confluency. So it's they form a monolayer, so shichva achat of cells, and then you come with a pipette and you scratch. So you create the experimentalists create an artificial wound here, and on the onset of the experiment, at the beginning of the experiment, the cells at the front, they fill the empty space, the cells at the back do not. And the question is how this information propagates from the front to the back. One of the possibilities is that it's like a stoplight, right? I mean, each cell sees the cells in front of it, and then when they're all squeezed together, and when some spaces freeze up, right? מתפנה, the cells are coming and moving to that direction. And I'll show you that it's not, as always with cells, it's not as simple as you would think. But before that, I mean, the standard ways to quantify monolayer migration assays, so this type of wound healing, it's highly used. It's used in a lot of papers. But basically, it's used as a measure to show if the cells are migrating faster or not in different treatments or different cells or whatever. This is the purpose. And in order to do that, in the old days, it was, you just take images and you see, oh, this is the starting time, this is the ending time of the experiment, and this works faster than this. You can take snapshots over time and see. In this case, you can see that B, for example, is converging faster, so they are migrating faster. The wound healing from two sides are converging faster, and so treatment B is faster than A in a wound healing. You can see it also a quantification here. But and when we look at the wound healing over time, so how the front is progressing over time. So the x-axis is time and the y-axis is space, how far the front is going. It looks very boring, like the front is moving in the constant speed. But when you zoom in at the beginning of the experiment, you see that at the beginning the cells are not moving, and then they are gradually starting and accelerating. And you can see it here when we look at the wound healing rate, which is the acceleration. You can see how nicely it's changed. So the derivative here, or the slope here, the change in the position, the healing. So the change in the speed of the monolayer front is increasing over time at the first 2 hours of the experiment. And the question is, what's going on in these 2 hours? What happens so the front changes its behavior? And the simplest hypothesis here is that the cells at the back are joining in the activity, the action. At the beginning, it's the front cells. They are the only ones that know where they're supposed to go. So it goes like slowly. And then cells from behind are joining in, and then it becomes more and more effective as a group behavior. Okay? So this is the motivation. When people want to look at what happens, to quantitatively look at what happens in the bulk of the data. So bulk is, I mean, somewhere in here. So what happens in the cell inside, not in the front, but inside. One of the standard approaches is called the particle image velocimetry. In computer vision, there are other methods with some other assumptions, which are called the optical flow, for example, where the idea is that you are looking at patterns, at texture patterns within an image in the next image, and you are looking at spatial correlation. You're maximizing for each patch. You're trying to maximize the spatial correlation, right? And when you find the maximum spatial correlation in the next frame, it means that you need to go to that direction. So there is a motion from this patch to another patch. If you have in cell monolayers, if you have trajectory of each cell, of course you do not need that. In this type of data, which is it's very hard to track the single cells. I mean, some of them are not very hard, but some of them are more. And especially if you want to do something very extensive, I had in this project thousands of movies, and I wanted to quantify the dynamics. And in this case, I'll show you, I didn't need the exact trajectories. Then this was much better at understanding what much more robust. So I'm less susceptible to mistakes. Tracking mistakes could be, could kill me. And so this was a way to avoid it using PIV, particle image velocimetry. So this is a results of PIV, I mean, visualization. You can start and you can see that it's kind of a grid. Yeah, I put a grid here, and for each grid, I look where it moves in the next frame, just based on correlation. You can get these errors. I mean, it's very, very simple. Okay, so I had here two projects which I'll share with you. Today I'll focus on one of them. So one, for the first project here from 2015 is how this communication is induced by local mechanical fluctuations. So basically, it's a more it's looking how single cells are affecting eventually the group, how information propagates across space and trying to coordinate the group of cells. And it involves some spatial clustering of group of cells that are migrating together. And another cool idea, which I might show because I talked today about reusing all data to make new discoveries. So this is one example of using that. I mean, everything here is data that was published already, which I looked from a different perspective. And the second, which I'm going to talk about today, is looking at what are molecular players driving long-range communication. And I mean, I'm going to show you some names of molecules here, but it doesn't matter. I mean, you don't need to know what the molecule X or Y for all you are concerned. I'll tell you what is the meaning, but for all you are concerned, I mean, it's X and Y. I mean, it's not, we don't care what are the molecules in this course. And one of the novelties here is a high-dimensional representation of spatial-temporal dynamics. And this was a paper published in 2017. And this is what I'm going to tell you today. So I'm going to talk about a group of proteins called the raw GTPases. Now, again, you don't need to understand anything about it. I'm just going to tell you why it's cool actually from an engineering perspective. So we have a group of 20 raw GTPases. 3 of them are super important. So the CDC42, RAC1, and RAW-A are proteins that are involved in many biological processes. These 20, and especially 3 proteins, are activated by 82 proteins called GEFs, guanine exchange factors. They are inhibited, so neutralim, by 67 GAP proteins, and they are affecting around 100 effectors. So if you think about it from an engineering perspective, why would 3 proteins need all of this control, right? So many activate. From an evolutionary perspective, think about why did the evolution create such a complex control machinery of 3 proteins? And the answer is specificity. So these are 3 proteins, but they're involved they are very important for many processes, cell biological processes, and they need to be controlled very precisely when and where in the cell to be turned on and off, in what context, and this needs a lot of control. So this is why the system is cool. And we try to follow how these GEFs affect raw GTPases, which affect effectors, which are affecting the things, a lot of mechanical, especially mechanical activities within the single cells, which are then translated to a collective behavior. So this is the pipelines that we're interested to see. And all the readouts that we'll use is the readouts at the collective behavior scale. So at the bottom here. So let me show you what I mean. So we started by just looking at the wound healing rate. What I told you earlier was just looking how fast the monolayer migrates. And this is what you see here is the difference between the knockdown, so taking out an activation of a protein, to the control experiment. And you can see that the red CDC42 and the RAC1, when they are inhibited, the migration is inhibited. So there is much less, cells are moving much less. Each dot here is an experiment, right? A movie. And when we inhibit RAW-A, you do not see this change. Although RAW-A is super important and important for contractility, kibbutz. The cell can generate forces through contractility. So it's a very important mechanism. And you would expect that when you reduce contractility, you will see reduced motility, but we didn't see it. This is just validation that it's not specific to one way of reducing RAW-A. Our collaborator actually used different ways to reduce the activity of RAW-A, and we didn't see any significant change in motility. So, okay, so we wanted to see what happens throughout the monolayer, not just at the front, but throughout everything, everywhere in the monolayer, going back into deeper cells. So what you see here is how we visualize that and also quantify, but I'll tell you in the next few slides how we quantify that. But first, visualize. So we created what is called a chemograph, which is basically a spatio-temporal representation in space and time of the dynamics here. So at the x-axis is time, the y-axis is location in relation to the monolayer front. So we had an algorithm that gave us the front. And then each bin here is in this case, it's a speed chemograph. Each bin here is the average speed of all the cells at a given distance, d, from the monolayer front at time t. Okay? So what does the diagonal means here? Anyone wants to try?
That's the shape of the cut, no? No, oh, no, it's actually, sorry, the location is different.
Yeah, so do you want to try more? So if you think about it, and I'll guide you through that, okay? So here you see at the top here, you see the cells at the front. At the bottom here, you see cells more at the back, right? I mean, it's not super back, it's like six rows inside, right? I mean, but it's still cells at the back. And you see that migration starts from the beginning, and cells at the back, it takes them much more time. It takes them about, I don't know, an hour and a half to start migrate. So basically cells at the start migrating from the beginning, and gradually cells from behind are joining in. This is what this diagonal means. So there is a front to back propagation of this migration properties. So this is a cool visual representation of what's going in within this system. Okay, so let's look how does it look? I mean, we have here the three raw GTPases. Each time we deplete one of them, and we see how like Tal said, we want, in order to learn something about the system in cell biology, what we usually do, we kill something, some transistor, and we see how the systems react, and then we say what is the effect of this transition. Okay?
So this is how you're going to find exactly which protein is doing the communication and tells everyone to move, everything to move.
Exactly. Yeah. I mean, it was luck, right? But eventually, this is what we found. So if you see here, we can see the CDC42 and RAC-A. Everything is, there is no red, right? So basically, all the migration is gone. It's expected. People know that it produces migration. But in the raw A, we see something different. If here it takes about 1.5 hours from the front to the back, the communication to propagate from the front to the back, we can see in the raw A, so we see red here and we see red here, and there is no significant difference in the overall motility, but you can see that here it takes much longer, and here the cells are synchronized. So when the cells at the beginning start migrate, the cells at deep within the monolayer also start migrate, which is pretty cool. So I want to visualize that, and if I stop somewhere around here, you're going to see the velocity maps that I told you through PIV, how they through particle image velocimetry. If we look at the control experiment, we can see that cells at the front are organized, they know what they're supposed to do at this stage, but cells at the back are completely confused. They are not migrating much, they do not know where they're supposed to go, they're all, you know, they're all confused, they don't have the information.
But one small question, how can you know that the protein that you kill is not affecting the actual move of the cells? I mean, maybe the cells need some protein in order to do the actual move, not to communicate. How can you eliminate this option?
Eliminate this option that it's just affecting
not this one.
Just creating the cells to just move, and then you'll see that they move, but not in a specific. In principle, you'll not see that they are moving to a specific direction. It will be a mess, right?
Okay. Okay.
Yeah, but it's a very good question, Gil. I mean, I'm very proud of you for asking that. It's a question that the biologists would ask, which is a good thing in my course.
I never learned any biology anyway.
But it's a way of thinking. It's not related to what is your background. Anyway, it's a good sign for curiosity, really, on what happens and why couldn't it be a different explanation. I mean, you don't believe me, which is good. Okay, so we found this initial cool observation, and once we had that, what we wanted to do is to check that systematically. So we wanted to go to the guest activators of that and test all of them very systematically. So here is what we did. We did a screen. We had 81 guests, but the screen was not like Ann Carpenter screens. It was much more validated. So each perturbation, it was very heavily validated by our experimentalist collaborators. So they worked really, really hard. Each time they depleted the proteins, they wanted to make sure that it was really depleted, and they used different way to deplete it, and it was a lot of experimental labor here is hidden behind these numbers. So 81 guests, three valid hairpins are different ways to deplete this protein to make sure that it's not a side effect of, you try to deplete something, you cause some side effects, and what you see is because of side effects. So if you deplete the same thing from three different ways, and you see the same thing, it's probably not a side effect, okay? We are looking at different locations. We're imaging different locations. Overall, we analyzed 3,000 videos. I had to design, I'm not going to tell you about that, but I had to design an image analysis pipeline that was robust because I couldn't do 3,000 movies. I couldn't do anything manually. So I wanted it to be fully automated and not be very sensitive to algorithmic faults, right? It needs to be exact. And the chemographs were really good at that. I had to deal with a lot of variability within the data. For example, day-to-day variability was a big issue here, but I'm not going to talk about it. And we wanted to define measures for screening. So this is how the screen was done. Started with 80 out of 81 guests. 75 of them were expressed in the cells because they are not necessarily all even active within the cells that you're using. So they checked that, and 75 out of the 80 were. And then they checked the knockdown efficiency. So PCR, you hear that from the corona perspective. But anyway, they knock down a protein. They want to make sure that the protein is really went down. And then some of them, 16 of the guests, we had knockdowns that was not very efficient, so we didn't follow up on that. And then we had the remaining 59 remaining guests, which we live image and did the actual experiment. Okay, so how do we encode spatial-temporal dynamics? So this was, remember, it was published in 2017. It started in 2014, I think, or 13. And it was before the days of the deep learning in cell biology. So not because now I would use that probably. But what I used before, I created those chemographs, and then I extracted features, basically just downsampling of this chemograph. So I had here 12 features, and it was robust. I could use other numbers as well. But the features were also, they had a very clear meaning, right? So looking at features 1, 2, 3, 4 is the acceleration of cells at the front over time. Looking at the 9, 10, 11, 12 are the acceleration of cells at the back. Features 1, 5, 9, for example, is a spatial derivative at a given time. So what are the changes between cells at the front and cells at the back at the beginning of the experiment? So this was the feature vector, and it had a very clear, they were not just numbers, we knew what are the relations between them. So once we had these numbers, I mean, now we're going to completely, we have a matrix now. These are the features, these are the experiments, these are just control experiments, and you can see how much the variabilities are even within the control. It's normalized, and this is our data. We can start playing with that. So one thing that I did for screening was doing a PCA, and then correlating the PCs to the wound healing rate, which is what everybody measures. So we see that PC1 measures the wound healing rate, PC2 and PC3 also has some correlation, but not exactly. And now what I wanted to do is to screen these 59 gaps based on these principal components. So I wanted to understand what each principal components mean biologically, and then use it for screening on the 59 gaps and see which gaps is affecting what. So now comes into place the fact that I know what are the features, and they have a very clear organization in space and time, right? So I looked at the coefficient. So basically PCA is just a linear transformation. So I could look at the linear transformation. How, when I get a feature vector, how do I transform it to PC1, to PC2, to PC3? And when looking at that, look what emerges. Basically, that these PCs are encoding a very, actually trivial, if I would think about it from the beginning, trivial properties within those chemographs. Now, it's not that trivial because the chemographs are much more balagan than what I showed you are the prettiest chemographs. In 3,000 chemographs, it's a lot of mess going on. So it was hard to, for my defense, it was hard to see that. But once I got this, it was clear. So if you think about it here, you get the main component here. You see all the features get a positive value about the same scale, which means all that we measure here is the magnitude. So how fast the cells are moving. Here you can see, so the first 4 features, right, are the cells at the front over time. The second 4, the middle cells over time, and the cells that are more at the back over time. So when I look at these patterns, I interpret that as a temporal derivative. So cells at the front get a positive value, cells at the back get negative values, and we look at basically we look at the differences between them. And the last PC is a spatial derivative. So cells at the front get positive values, cells at the middle get zeros, and cells at the back get negative values. So this is encoding a spatial derivative. So now, from a data-driven perspective, not like a physicist to define from the beginning what are the properties, I let the data tell me what to measure. But now I can assess, I can say that the variability within the data, and when you look at this diagonal, it actually makes a lot of sense. It's a magnitude, temporal derivative, and spatial derivative, and I can use the principal components for screening. So how does it look for the CDC42 rack 1 and row A? Basically, it looks, these are artifacts. I'm not going to tell you why, because I don't have time. And artifacts because of because it measures something else. It measures the change. The change in PC1 explains the change in PC3 in the green and the red. But what you see in the blue is that there is no change in the speed, like I showed you before, but there is a change in PC3, which is the front to back propagation. So that's the phenotype. And here you can see that this is not one experiment, but this is the quantification. So it replicates across many experiments, as you can see here. So we did the same things that we did for speed, we did for directionality. So cells are moving into the direction of the wound edge, and we got the same thing. Screening methodology. I just want to show you, maybe I'll show you just a few things. So this is the wound healing rate of the controls. Look at the variability. Control cells, so basically these are the same cells. Some of them are moving 20, some experiments are moving 20 micron per hour, some of them are moving 60 micron per hour, 3 times faster. With this variability, it would be very hard to say that something is an extreme here, right? And this is why when we do screens, we measure and we have controls in every day, because a lot of this variability is explained by the day. The cells are feeling better, are feeling worse, and they act in the same way. So we want to exclude the possibilities that the day is what we get here. So what we do in each experiment, we have this P super, which is a control experiment, and we use the different hairpins in this case. So we have a control for each experiment, which we can see if the experiment is changing in relation to that day's control and not compared to all the data. What is a phenotype? How do we define if we found something interesting or not? We're looking at the control experiments, and we're looking at our condition, something that was changed. And we're looking at two parameters. First, how close are each experimental observations to one another? And this means that the clusters are, as the clusters are more צמודים אחד לשני, capture are closer to one another in this space, in whatever measurement, and they are distant to one another in the clusters, this makes a better clustering analysis measure, right? So we use different measures for that, 3 different measures to define how good is the separation here between those two. Okay, how would we validate what we get? So what we wanted in the experiment, we also want to be able to say that our measurements make sense. We want to make sure that they really make sense. So the way to do that, we could look at this CDC42 and RAC1, which we knew had a major effect, and also beta-PIX, which is another protein, which we knew had a good effect, and we use them as positive control. So for each experiment, we can see that they have an effect on migration. When you reduce these proteins, we have an effect of migration. And then by chance, some of the experiments just didn't work. So 0% knockdown means that the experiment didn't work. So we weren't able to actually reduce the activity of this protein. So in principle, this should behave similar to control cells. But since we still did there something, we might have side effects. So we tried to reduce protein A and we didn't reduce protein A, but maybe by doing what we did, we reduced other proteins, and this were used as off-target control. So maybe basically measuring side effects. If we see something here, it means that there are side effects in our data. This is what we get when we look at the whole screen. So these were the positive controls. You see, okay, you see a big change, right? You can see big change, which means that we find something. And when we look at the knockdowns at 0 and knockdown over 50%, we see that most of the data is clustered around the same thing, which makes sense because in screening, most of our data is going to look like a control. This means that these experiments are similar to the control. And in the screen, we're not interested in what looks like the control, but we're looking for the outliers, what look like not like a control. Okay, so this is what we're looking at. Yeah, and these are hits. These are false positives because here we are not expecting to see a phenotype. So if we see here something, it's because there is a side effect, or we're measuring error because of noise. And this would be false negative because here we expect to see a phenotype each and every time, but we do not, of course. It's a very noisy thing. So here is our screen. And what you see here are different GEFs. So each color is a different treatment. The blue are just all the rest of the molecules. So I should have here 59 colors or even more. And I just put here, everybody is a blue. And the ones that are interesting, I color coded them. So the positive controls, which we know have an effect, are here. Look how noisy it is. I mean, even in the positive control, there are features and experiments where I didn't get the phenotype. Okay, and then when I get something, I want to validate that. I mean, these are 10 standard deviations, so it's a lot. But yeah, I mean, you can see that it's very, very noisy, and we need a lot of replications to actually say that something is occurring here. So after we did a lot of, we found some interesting hits, and we replicated, my collaborators replicated experiment a lot of time, and we find the interesting hits that are specific raw GTPases, and then we wanted to follow up on them. And you can visualize these hits and see how they change. So some of them are boring, some of them are, all that they do is reduce the speed or reduce the directionality, which is less boring. And some of them has an effect that's similar to the raw A. For example, RHGF18, you can see here the diagonal versus the straight line, right? In the communication. But for example, in when you down-regulate RHGF11, you can see that in the control you have very little directionality, and in the same day, when you reduce ROI, you get more directionality. So the cells know where they're supposed to go. Yeah. Yeah, I'm thinking whether to continue here or wait to next lesson. Okay, I'll go quickly. You know what? No, I'll start next time because there are some interesting things on the follow-up which I want to. I think are. Okay, you know what? I'll wrap up. I'll wrap it up.
I tried to ask something, but I was muted. How exactly do you measure directionality? I mean, you see things move, you see things that are not moving on the right direction or the direction that you expect. So, I mean, how would you give them numbers? That it's
yeah. So remember that we're looking about chemographs.
Yes.
In here is the let me go back for that. That's worth a while. It's each bin in the chemograph is the average whatever I measure for all these cells in a specific time.
Yes.
Directionality would be the vector here. What is the direction of this vector, right? So if everybody are going and the right direction is toward the wound. Okay. So I mean, in principle,
it just sounds to me something which is very hard to specify or to measure.
Well,
because
I mean, basically it was just so you know. Maybe I'll paint a little bit. I'll color it a little bit. I mean, if you see vector like this was a high directionality, and if you see something like this, it's a low directionality, right?
Mm-hmm.
And basically it's you can look when you look at something like this, and the direction is this direction, the main direction, right? So you can look at the dx dy, right? And let's say the dx is the dominant direction. And you can do just you can just look at the relation between those two, and based on that define, and then do it for all the cells here, and then take the number and put it here. So yeah, I mean it's pretty standard. It's not, there is nothing.
Okay. It's just I mean when I'm trying to think how to do it, it sounds I mean, how to really take the numbers, but okay.
Yeah. So. Yeah, I'll.
It's basically was done. All those numbers were taken with what you wrote for the image processing, I guess. The image processing that you did gave you those numbers, basically.
Yeah. So basically, if you think about the pipeline, there is the basic image processing pipeline, which is identifying the front, right? Automatically identifying the front and tracking it over time. This is one thing. Second thing is doing the PIV, looking for each patch within the image and defining to the next time frame where to define the direction. The third step is building the chemograph. So going in steps over space and time, and for whatever I'm interested in, migration or directionality in this case, take all the cells within this band at a specific time, calculate the mean, and put it into the chemograph. So this is the basic image analysis process. And usually when people do chemographs, they stop here. For me, it was the beginning, because for me, this was a spatial-temporal representation of a whole experiment. So with this representation of chemographs, this describes a full video with one image. The dynamics in space and time of everything that happens within the, not everything, but as much as I could based on this, within an experiment with this one image.
Yeah.
So this is a very, very condensed, it has a very condensed information within it. and now I use that as my data. So now and today you could do it with neural networks. You can use these and put these as images into neural networks and let the network learn the representation. I did it more directly by downsampling and using these as engineered features, trying to understand what they mean, going to the biology, trying to understand what's going on, finding the phenotypes based on the screening, and then zooming in and going and trying to understand the deeper what's going on here. So I think I'll skip and I'll not go into details. I'll just go to the bottom line. But the idea is here is that we looked at and we talked about networks or protein networks. Tal talked about it a little bit. And this is what eventually this is our findings. Okay, this is what we discovered. So this is there are some methodological novelty here. I mean, all these methods that I described, but eventually we use it not for the sake of just having a tool, but also to learn something new about the system. And this is what I wanted to highlight here. And I'm going to and everything is based on these measurements that we do. So here is what we found. We found something that is kind of, oops, no. Okay, so first we find something that was quite boring, this SOS1 and RAS pathway, which basically they control speed and directionality. So if you kill them, you are going to reduce speed and directionality. This is boring. I mean, this you can do by just measuring the modular front, and it was already known. I mean, there is nothing special here. What was special here, that we went in the pathway up to the activator of the ROA and down to the downstream analysis of ROA, what ROA does. And ROA is involved in contractility again, in קיבוצים של התא, יצירת כוחות בתאים. And we found here that there is a different effects here. And what you see in קווים מקווקווים is what is new, what was not known before. Okay, the relations that were not known before. So we know that GEF-H1 and ArchGEF-18 are activating ROA. We find here that these are also activating ROA in the context of restraining long-range communication. So if you reduce ROA, you're going to get better long-range communication. Okay, so ROA is restraining long-range communication, and the TROC, which is something similar to ROA, has a different effect because it is restraining long-range communication. It is also restraining speed. So if you reduce ROC, you're going to get your cells actually moving also faster. Okay, and yeah, so this is ROC, and you see it has a different effect, and it is different activators. And then we found that it goes down to the contractility machinery. So if we reduce contractility, which is counterintuitive, you reduce the contractility, את היכולת של התא לייצר כוחות, and you get faster front to back propagation of information. So it's not exactly true here, because if you reduce a lot of contractility, the cells are just going to be like smartutim. They cannot do anything. If they cannot contract and generate forces, כבר לא שווים כלום, and they cannot move. But if you reduce contractility only a little bit, the cells can still generate forces on the one hand, but on the other hand, they can also, they are less stubborn. They are more able to listen to other cells. So think about the cells that is super contractile. אני שרירי כזה, אתה מנסה להגיד לי, אסף, בוא לכאן, come here, and I'm saying, no, I'm very contractile, אני מאוד חזק ולא מקשיב. If you reduce contractility a little bit, I can still walk very effectively. But if someone tells me, Asaf, maybe you should come this direction, I'd say, oh yeah, probably I should go, and I go. And this is what we found here. So there is a fine balance, and these systems are all about balance, right? They are controlled very precisely. There is a fine balance between the ability to generate motile forces through contractility, and the ability to be guided by mechanics, by telling you should go here or there. Yeah, so here are the paper. There is a paper, and there is actually now Yeshaya from the lab he generated in the summer pipeline. And MATLAB, it's just an old because my postdoc lab was working in MATLAB. Now all my lab is working in Python, but it was kind of a legacy code of me. And Yeshaya decided to do it, used it and created from it a book chapter for people so they can use it more freely. So these were the people that were involved in this study. Actually, Alan Hall is the biologist that led the experimental side. He was from he was a British guy from Memorial Sloan Kettering Cancer Center in New York. He tragically died actually during this project. He was 60 something. He went jogging in Central Park and he just fell and got a heart attack. And then Mike Oberholzer joined in and helped us finish it. And Yanu did most of the experiments, and Angeles and Shefali helped her finish the experiments. And yeah, Gaudens was my boss there. I mean, I was not here yet, so he was the boss from my side, my boss. I know that I skipped a few things, but I think that I wanted to show you a few things about screening and all the difficulties that come into that. You need a very robust pipeline that will work well for a lot of data. You need to be very careful about different aspects of day-to-day variability, off-target effects, all kinds of crap that happens in this super complex biology. I mean, you are not going to get any more real-world data and variability and difficulties than that. And not just building the tools and doing the screening, but doing the extra step of, and this is why I picked the paper from me, because I know that exactly. In other papers, I do not necessarily know all the details, that we can learn something new about the biology. So now we learn something new about how these processes are balanced. And you can think about it that in some situations, you want the cells to be more contractile and more stubborn. And in some situations, you want them to be more flexible in making decisions. And this could be controlled with this way. Okay, so I think that's it. I'm going to write it, so you're only 5 who are brave enough to stay to this time, and 2 of you are the speakers of today, so you're just feeling uncomfortable and staying maybe. But anyway, I'm going to write a message in the, I don't care, as long as I have one person who listens to me, I'm fine. And actually people from all around the world are asking for the recording, so I don't know, I mean, maybe people are watching. I didn't even check how many recording views are there. So anyway, I'm comforting myself. Anyways, I'm going to write a message in the Moodle, and I'm going probably to talk about it next week as well, about filling up the survey, Mashuvim. So please, I mean, it's very important, and I use it. I can tell you, maybe next week I'll tell you about what I did following the feedbacks that I got from last year's survey. So I think I improved the course a lot thanks to that. And I understood what worked well, what was good, and what was could be improved. And I always want to improve. So I would appreciate if you would feel it. You need to know that I'm not getting. I'm knowing about the results only after I submit the grades. If you didn't know that, so there is no way that you're going to say Assaf is a crappy lecture or something, and I'm going to להתנקם בכם או משהו כזה. I don't know. So I don't know. Probably you don't care, but my first year students, I seem to care about it when I told them about it. טוב, אז יאללה, אז see you